The overall goal of the following experiment is to perform in vitro dynamic clamp in cortical neurons using the freely available software toolbox LCG. This is achieved by preparing slices of rat somatosensory cortex for patch clamp recordings from the pyramidal cells. As a second step, a series of automated electrophysiological protocols is applied to the recorded cell, which provides a standard characterization of the cell that will be useful for subsequent analyses and comparisons across cell types. Next, the injection of excitatory and inhibitory synaptic events is simulated in order to recreate the high conductance state experienced by the neocortical neurons in vivo. The results show how background synaptic injection modulates the gain of the cortical cells.
The main advantage of this technique over existing methods is the possibility of embedding experimental recordings into more complicated workflows by using, for instance, the result of one experimental protocol as the input to another protocol.
This method can help standardize electrophysiological protocols, accelerate data analysis, and pave the way for data sharing across laboratories.
Though this method was originally developed for whole-cell patch clamp recordings in vitro, it can also be conveniently used for other experimental preparation and protocols, such as in vivo patch clamp recordings or extracellular recordings with microelectrode arrays.
After extracting the rat brain, discard the cerebellum and separate the two hemispheres along the midline using a scalpel. Remove excess fluid from one of the hemispheres and glue it on an inclined platform with a drop.